a minimal viable product for a cultivated meat company has to, first of all, match or exceed the taste and texture of conventional meat. Second, it needs to reduce the cost of the finished product. And then finally, it has to immensely scale that production to even have a small fraction of the conventional meat market that we talked about, as well as in what you talked about on your previous podcast in December with Beat Thalman.
Hey, smart scientists. Welcome to another episode on the Smart Biotech Scientist podcast. Do you think cultivated meat is a viable alternative? Let's find out with our guest, Stephen Lang, who is a biopharmaceutical leader with 20 years of experience in discovery to development, process improvement, and financial and strategic planning. Stay tuned. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Stephen, on the Smart Biotech Scientist podcast.
Thanks for having me. It's been my pleasure.
Yes, pleasure likewise. It's so good to have you on the show today. Stephen, share something that you believe about bioprocess development that most people disagree with.
That's a great question to start with. And trying to think of controversial ideas that I have, but I think it's rather vanilla. And I think the biggest problem facing cultivated meat is socioeconomic. It is not the science. So I have great confidence in our ability to continue the 40 plus years of biopharmaceutical trajectory to solve some of the scientific challenges to produce safe and tasty cultivated meat. However, I'm not so certain that we have the societal or economic leadership necessary to make it a reality. So that is where my concern is, and I think that's probably something that maybe some people would disagree with.
Before we start talking about our main topic, cultivated meat production, I'm curious, Stephen, what led you to start in biotech and how did you end up where you are at the moment?
It has been a very fascinating road, and it started out when I was young. Back when I was in, I would say, 7th or 8th grade, my father, who was keeping up with topics at the time, said to me in a very offhand way that he thought that genetics had a very interesting future. And for me, for some reason, that really resonated. And after that point, I really focused all of my education on learning more about genetics and molecular biology in particular. And I kept that focus all the way up through graduate school, at which point I had a molecular microbiology degree. It wasn't until my first postdoc that I kind of got my head up out of the weeds of molecular biology and started working on my Master's of Business Administration, an MBA. And this was mind-blowing for me because it made me realize that there are people out there that have studied organizational behavior, management, and leadership to the same depth that I've been studying cellular signaling pathways. And that was an eye-opener for me. And I'd have to say that, the reason I got started in my MBA was for two reasons. First, I've had experiences both in academics and in industry that showed me that great scientists don't necessarily make great managers or leaders. And a second reason I wanted to work on an MBA was I understood that scientific endeavors like what we're trying to do in medicine and in the food industry really requires a huge collaboration of a lot of different type of scientists. And I wanted to learn the skills necessary to help amplify my impact by helping other scientists be able to collaborate. So as you know, it takes, much more than a village to get a drug to the market. And we're also seeing that in the cultivated meat industry is that it takes a huge collection of people. And getting those people focused and directed in the correct way is going to be paramount for us to be successful. So I had some really cool and I would say interesting key moments in my career that kind of led me to where I'm at now. One of them was first in the very beginning. My first job outside of academics was at Centocor, which was the biologics arm of Johnson & Johnson. And I was asked to work with a team of leaders who were charged with reducing the cost of the active pharmaceutical ingredients, API, the COGS, cost of goods sold, from $1,000 per gram to $100 per gram. That was a huge challenge for us. And we had to interrogate every part of the critical path from the cell lines all the way out to the final product. And from that experience, I really learned the value of the critical path and its relationship to the critical quality attributes and the costs that are ultimately passed on to the consumers or the patients. The other thing that I learned while I was at J&J, they have a credo. And their credo is essentially a business vision that says that the consumer or the patients are paramount and the employees are secondary. The stockholders are, very last in line when they're making business decisions. So that was really influential to me because it allowed me to build a philosophy and kind of a approach to my science to keep the product and the consumer in mind first and foremost, and then the employees who are helping us do the science second most. And if you can get those two things correct and well functioning, then you're going to be successful. The other inflection in my career was when I was a technician between undergraduate and graduate school at the University of Penn and in the lab of Professor Fevzi Doldal. It was a very international lab. In fact, I was the only person in the lab of 15 or 12 to 15 people that was born in the United States. So we had a lot of diversity and a lot of communication channels or challenges. So it was a lot of fun to be able to learn the value of having a diversity of background and disciplines as well as cultural backgrounds to help solve problems in a very diverse way. So those were the big things that really kind of led me to where I'm at now. And those you'll see in the later parts of the conversation that it kind of comes all together.
So that's an exciting journey, Stephen. So my question is now, why did you transition into cultured meat?
So that maybe also a little bit of a long-winded explanation. First and foremost, it was a technical challenge. It was really the idea of taking what we know of biomanufacturing as a very expensive process. It inherently has to be expensive. And using that same technology to build a process that can produce a food commodity and a very low profit margin. So the absurdity of that certainly made it interesting. It also helped that I had colleagues that I had a lot of respect for already made the jump into cultivated meat, like Gianti Laccaretti, Bob Kiss, and Kevin Kaiser. All of those were huge, influential people that I really respected and were already working in the space. The other thing is, as I further dug into it, it became even more intriguing because this is a multifaceted problem. Cultivated meat is like a great murder mystery where every clue leads to another uncertainty and everything seems to be connected. And in cultivated meat, the challenges that we have that we're really facing span not just technical, but it's also business challenges, political, economic, cultural, and finally environmental. And all of those things are interconnected. So pulling one string on an economic problem may have an environmental or a business problem that you have to solve for later. So that is a fun challenge to take. The other part of it was this is a little bit of an extension from my time at Centocor when we were reducing the cost of active pharmaceutical ingredients from $1,000 to $100 a gram. Now with cultivated meat, it's 2 orders or maybe even 3 orders of magnitude greater of a decrease in COGS. So we're trying to get the biomass cost down to pennies on the gram rather than hundreds of dollars per gram.
Do you think cultivated meat is a viable alternative for our food and feed system?
Honestly, David, not quite yet. I think we're going to get there, but we need to have some more time. If you take a step back and really kind of think about it strategically, a minimal viable product for a cultivated meat company has to, first of all, match or exceed the taste and texture of conventional meat. Second, it needs to reduce the cost of the finished product. And then finally, it has to immensely scale that production to even have a small fraction of the conventional meat market that we talked about, as well as in what you talked about on your previous podcast in December with Beat Thalman. So those minimal viable product characteristics assumes a lot. And it also assumes that there's going to be a market demand for cultivated meat, which is more of a socioeconomic question that is being influenced currently by politicians who are interested in banning cultivated meat to support industrial animal farming as well as meat processors. And then finally, this minimal viable product assumes that cultivated animal cells can recapitulate conventional meat structures, aromas, and taste without extensive additives and processing like plant proteins or flavors or texture-inducing processes. So I'm certain that we're going to crack this nut, and we will find solutions that will allow us to be more efficient at building protein for human consumption than what current conventional meat processes are. And there have been numerous life cycle assessments of the cultivated meat industry that demonstrate that through cultivators, we can essentially be more efficient taking feedstock and turn it into high-value protein than animals can.
Now, a question I ask myself often is cultivated meat a bubble that eventually will burst, like similar to the tech bubble in the early 2000s?
Yeah. So, well, certainly venture capitalists have invested more than $3 billion into cultivated meat, and there are currently no significant products on the market. So that sounds like a bubble to me because the VCs will be looking for an exit in the coming years. The horizon for venture capitalists are typically less than 10 years. And I think that for the cultivated meat industry, it's going to take us longer. And as you know, David, in 2023, we saw some retrenchment and consolidation of the industry as companies have failed, either due to lack of funding or the science just did not pan out. And I expect that the companies that survive will have a combination of great innovators and technology that leads to patent protection and excellent leadership that understands the messy path of scientific innovation while keeping good stewardship of the finances and the brand going forward. So that is what is critical to survive long enough to create a market demand and a superior product in this novel industry. And as I was saying before, it takes a village. It really does take a lot of different scientists and a lot of different disciplines to be able to deliver on what we ultimately think cultivated meat can help us in all of these problems.
So, Steve, let's look at the technical sides now, because yes, I mean, there is a tremendous challenge. You said we need a lot of people on board. We need the funding. There's much complexity. Can you tell us what the current challenges are in developing and then scaling this process up at a massive scale so that we can deliver cultivated meat that tastes good and is still not too expensive?
That's a great question, and it's a big thing that a lot of people focus on, but it is really only part of the problem. So speaking in vague terms, and maybe to help your audience appreciate really what we're up against, the challenge is to produce billions of kilos of meat in a cost-effective process. So if you think about downstream processing of cultivated meat, after harvesting the biomass from a cultivator, it is handed to a team of food scientists who are going to then formulate that into a final product. To be effective, a food scientist really wants to have something that's similar to a meat block and to work with, because they want to basically take something that has the flavor, the texture, and then just add a little bit and put it in the right shape and form to produce a product. When you think about that, and anyone who's run bioreactors know what a packed cell pellet looks like, and that's what we're kind of thinking about when we think about a meat block. And we also measure cell density in packed cell volume, so PCV. And typically in 1% packed cell volume, there's about 5 to 6 million cells. But let's assume the technology really advances and we can get to a point where we're generating tissue or suspension cells at 50% packed cell volume. That means that 50% of your media is going to be cells. So 500 grams of a liter will be biomass. And if you had a 100,000 liter bioreactor and it took 1 week to produce that amount of cells, then you would get essentially 500 grams per liter. And assuming that 500 grams per liter out of that 100,000 liter bioreactor didn't need to be diluted and could go straight to the market, that's a great place to start. According to the FOA, the world meat production is about 6 billion kilos per week. So if we were to capture 1% of that market, we would need to make processes and infrastructure that could deliver on 60 million kilos per week. And to make 600 million kilos of cultivated meat, you would need about 12,000 of those 100,000 liter bioreactors to work 24/7 all the time. So that is really the scope and size of the challenge that we're working towards really kind of figuring out. And with that, it's not so much the scaling as much as understanding the science, as well as the cell biology, the nutritional loading, as well as intensification. All of these things that we've been talking about within the biopharmaceutical sector for a very long time. And the fun part is that very smart people like to have or are motivated by such absurd challenges. And as you know, recent biotech has been really going towards smaller scales with personalized medicines such as gene and cell therapy, as well as in more potent biologics. So, the days of 25,000 liter cell culture bioreactors for biopharmaceuticals may be numbered. So a lot of that innovation is going the opposite direction to what the cultivated meat companies really need. They need those scales or economies of scales to demonstrate that they can deliver on even a 1% part of the total meat global demand. So several companies of the cultivated meat companies have indicated that they're going to be building infrastructure of 50,000 liter or beyond in bioreactors. That scale hasn't been achieved to my knowledge, of animal cell culture. So that's going to require extensive research as well as collaboration between cell line development, media, process, analytics, mechanical engineers, sensory scientists, tissue engineers. The list just goes on. And then maybe just to finish out, hitting those cost targets are going to be paramount to being able to deliver on the promises of cultivated meat. And those cost challenges really come down to the price of the raw materials going in, the process efficiency, and then, of course, the operational efficiency. And all of that will require innovation in each of those fields to be able to drive down the cost to become parity with conventional meat.
Yeah, so unlike a biofarmer in the cultivated meat, we are going towards large volumes and we have a tremendous cost pressure because we don't have these huge margins we still have in the biopharma. So I totally agree with you. We need innovations. Can you tell us a few specific innovations we will need to get there?
Specific innovations? Well, certainly advances in cell engineering. We need to be able to figure out the connection between the critical quality attributes for a cultivated meat, which is going to be sensory attributes and nutritional attributes. We need to understand the basic biology is cell signaling, if you will, that's leading between those phenotypes and the cellular mechanisms that we can then pull those levers to increase, say, for example, the amount of umami that's retained in a biomass so that we can hit those consumer preferences that we really need to hit. The other thing is, as we talked about, just efficiency. How can we take from feedstocks, raw materials, efficiently convert that into biomass that will be then consumed as energy? That is another place where engineering and process engineering will come in play that will need to really drive the innovation in order to hit some of those targets that we're going after. Those are just two examples, but there are many more.
If we produce at that scale, we need also huge quantities of media. I think that's another challenge. How do we get those huge volumes at a consistent quality?
Great. Yeah, the supply chain is going to be really another important part of the cultivated meat equation. And cellular agriculture promises to use more media than what is being produced today for all of biotech. And so that is a significant challenge that we need to explore alternative raw materials. And unfortunately, we all know about the sensitivity of animal cell culture to even low-level impurities or changes in media components. For example, divalent cations impact on glycosylation, as you talked about in your podcast with Thomas Rexer. So to be able to drive down the media costs, it's going to be really important to substitute in cheaper and larger volume alternative raw materials and build those raw materials into the process as early as possible so that you can demonstrate safety, consistency, and appropriate sensory CQAs. So this will require not only, a supply chain that is really out there looking for high quality alternative raw materials at a lower cost, but it will also involve then building quality systems and the analytics to really make sure that we're delivering consistent raw materials as inputs into the manufacturing process.
Hey, smart biotech scientists. I hope you've enjoyed our conversation with Stephen Lang. In part 2, we are continuing our journey and we're talking about the sensory complexity, about analytics, and the regulatory landscape. And perhaps you are not sure how you can develop a robust, scalable, and cost-efficient production process. So I want to offer you a free 30-minute call to help you get started. Book your call using the link down in the show notes. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.